Hi everybody, I'm Vincent Racaniello for the American Society for Microbiology. I'm coming to you from Boston University. I'm at the Emerging Infectious Diseases A to Z meeting in celebration of the NEIDL Laboratories, the National Emerging Infectious Diseases Laboratory here in Boston. And I've taken the opportunity at this meeting to latch on to a few of the speakers and find out a little bit about what it is they're doing. My guest now is from Cambridge University in the UK, Ian Goodfellow. Thanks for coming.
How are you?
I know you've spent a long time working on noroviruses, right?
Okay.
The cruise ship virus.
Absolutely.
And I want to know what compelled you to go to Western Africa and do something with Ebola. What was the initiative for that and what did you do?
I think, like many virologists, I've been watching the epidemic, from afar and seeing it getting worse and worse and sort of waiting for the international community to respond. And you sit there and you think, why isn't anyone doing anything? This epidemic isn't like anything we've ever seen before. Why aren't people responding? And it just came a point where I felt that I had a responsibility, and myself and other academic virologists, we could provide some sort of support, certainly in the form of diagnostics. So it came out from a feeling that I had a responsibility to at least try to offer some support. But that said, it wasn't easy because most people I spoke to didn't want my help.
So who did you speak to? People in Western Africa?
So I, emailed all the various NGOs that were operating, and none of them said, unless you were a doctor, a medical doctor, they weren't interested.
Wow.
So it took me from July until November 2014 to find a way to actually offer some sort of help. And then that's where I got in contact with Public Health England. And they were, at that time, deploying people to West Africa. And I started working with them. So
you left your laboratory. The plan was to go for how long?
Initially, it was 5 weeks. I think the initial plan was the UK government were deploying teams of 10 to 15 scientists to run 3 different diagnostic labs, and we'd rotate out every 5 weeks.
I see.
So I planned to go once for 5 weeks, and I was there for 6 months or longer in the end.
Wow.
For various stints.
So the first time you just went yourself, you got on a plane and went. Probably took a long time to get there, I guess.
Well, the first time I went as a team leader with Public Health England. We had to go in and set up a diagnostic laboratory in Makeni. So at that time, Makeni was a real hotspot for Ebola transmission, and there were no other diagnostic labs in the district. So we were the first team in. Okay. We had to set up the laboratory. There wasn't, they built the outside of the lab and get the workflow operational.
Did you have any training before you went by anyone?
5 days. So we had training from Public Health England and Portadown, which was really good. But it sort of relied on, you rely on a lot of your basic skills you've picked up over the years as a virologist, knowledge of basically how to run a laboratory and how to set things up. So I have to say, it took us longer than I had hoped to set the lab up. It took 2 weeks before we were operational. So
you had a space that they gave you, and you had to bring machinery in, equipment?
So we had, the UK government had built this big treatment centre in Makeni, and actually the treatment centre hadn't been finished, but the laboratory shell was ready. So we went in, we got benching, we got all the equipment was shipped over, but a lot of the pieces of equipment were missing.
These are coming from the UK, right?
It was all being flown in for the UK. Stuff would turn up. We had to physically unload everything ourselves. So you had 10, you can imagine 10 British lab scientists carrying tons, literally tons of stuff ourselves around this building site, unpacking it all and trying to put it together. And in some instances, we had to actually go and buy stuff from the local market and make things and then put them into the isolators to work with. So.
So you were building diagnostic capacity. How long did it take to do that?
I think it took us about 2 weeks to get the lab operational by the time all the equipment turned up and we had the workflow running. So we opened on the 13th of December 2014, which was, much later than we'd hoped. But actually during that time, that was kind of when I started to think about the other things that we might be able to do. How could I either myself or my collaborators back in the UK provide support. And that's where the idea of doing genetic sequencing of Ebola virus.
Mm-hmm. So initially you set up PCR
Yeah.
diagnostics, basically. And did you start to get samples in?
So we would get samples every day, you know, up to 100 samples per day. Maybe early on, 30 to 50% of those were positive.
Mm. And did you handle those under BSL-3 containment, or were they inactivated first?
It was kind of, it would be barely sort of BSL-2 really, if you think about the laboratory, but it was kind of an enhanced CL3. I mean, strictly speaking, that's what it was designed. We had what are called flexible film isolators. So all the samples would go into the isolator and we'd work in the glove box, inactivate them, and bring them out and do manual RNA extraction like you would do.
Yeah, yeah.
And then just qPCR.
So you would do a PCR and then report the results to someone. And did you repeat them to make sure they were
Generally,
positive
no.
once?
I mean, just right back at the start, you know, it's a single, it's not even double technical repeats of the assay. Certainly near the tail end of the epidemic, if a sample came up positive, it would often be that you'd rerun that sample just to be 100% confident. Certainly with the flare-up, so, you know, if you consider a case was identified in January 2016, you know, they ran that sample 4 times. They had to be 100% confident that was positive before they said it was positive.
Yeah. So I did a TWIV in Glasgow, I think last year, with someone who worked in one of these PCR units, Gillian.
Yes,
Does it ring a bell?
Yeah, she was in Kerrytown, so they were the very first team out, I think.
Okay, so at some point you started thinking about sequencing the PCR products, is that right?
Well, we started thinking about sequencing of the full genome. So, you know, how do we, at that time, there wasn't much in the way of data. So the Broad Institute had done some. We'd done a very good job of getting those samples out and doing the analysis and releasing that data quickly. But it was very difficult, and there was a big bottleneck from the sample being taken to the data being available to the scientific community. So we thought if we moved the sequencer to the treatment center, to a tent next door, we could bring that time down and then release the data. And in some ways, it was good that I had never done this before because I was naive enough to think it would be easy. I hadn't really understood how difficult it was going to be. But so I agreed to do it. And myself and my postdoc, then a few months later, brought a sequencer to the tent next door.
From the UK, right?
From the UK, yeah. We got funding from the Wellcome Trust to do it, and we just processed samples.
Was that hard to get the sequencer?
You know, it was one of the hardest things I've ever done because you know, doing sequencing in a laboratory in a nice lab is relatively easy these days. You know, it's technically easy. But in that environment, you need everything, absolutely everything, from every single tip to every single tube to a chair, a bench, you know, racks to put the tubes in, the right rotors. So I spent a lot of time thinking about everything you need and what happens if one centrifuge breaks. You know, you need ultra pure water. How do you make ultra pure water in a tent in West Africa? So trying to piece all those things together, but it worked.
Yeah. So you started generating whole genome sequences.
Whole genome sequences, and we could do it within 24 hours. So we would get a sample, we'd produce sequence, and certainly from July 2015, we were a key component of the outbreak. So we were then taking that data, sending it back to the UK for analysis. Yeah. And then we'd have a meeting, a teleconference with the epidemiologists to interpret the data, and we'd write a report.
So the RNA that you used for this, did they come from serum from patients?
Yeah, from the plasma from the patients. The samples would be extracted in the diagnostic labs and then given to us, and then we just sequenced.
So what fraction of positive patients from which you could get entire genomes? Was it all of them, or?
I think we did pretty well. I would say we processed every single sample that anyone would give us. So we had 1,200 samples in the end we processed. And from those, we got more than 50% gave us full genomes. So somewhere in the region of 600, over 600 full genomes we produced from our little tent.
And these are from which country? Was it just where you were, or surrounding countries as well?
Mostly Sierra Leone. really later on in the epidemic, we were sharing samples with people in Guinea. So there was an ongoing cluster in the border districts between Guinea and Sierra Leone. So we would share samples, and in Guinea they were doing sequencing by the MinION.
Yeah.
And we would compare the data, we would share samples and make sure that we were getting the same interpretation.
So over 600 full-length genomes. Is that more than the Broad study?
It's more than, we did a third of all the sequencing of all the Ebola virus genomes that came out of that entire epidemic.
Yeah.
And I say we, it wasn't just me. Of course, I did a significant amount of that work, but it was me, people from the Sanger Center. We had a small team. It was typically no more than 2 people at any one time. From my laboratory and from the Sanger Center, we'd rotate out every month. More people would go out and just work non-stop for 5 weeks.
Yeah, and all that data is out there now? It's in a database and anyone can access it?
Yeah,
Have you gone through it and looked for things? Have you published anything from it?
I keep getting asked these questions. You know, I work on noroviruses.
Yes.
Okay, the positive sense RNA virus. I've only ever worked on positive sense RNA viruses. I used to work on polio and enteroviruses. I don't really understand negative sense RNA viruses, so I'm not the best person to interpret that. So my approach was very much produce the data and give it to the people who can understand this.
That makes perfect sense. I mean, you wanted to go there in the first place to help out, and so this is not your virus, so you just give the data to people who are going to use it.
For me, it made more sense. I think it's a faster route than to me trying to learn all this stuff. And you know, there are people much better placed who can interpret this data than I ever will.
No, for sure. It's interesting. Now, how long would it take to generate a full-length genome on the sequencing machine? You said a day, roughly?
It would take a day, so 24 hours if we get the sample in a reasonable time, you know, in a reasonable time of day, from us getting nucleic acid to getting assembled genome that we could analyze. And that, you know, to do that even in the UK or in the US, that's difficult. You know, from a clinical sample, 24 hours. But we were doing that in it was, you know, an insect-filled tent, I call it. It was a nice tent.
It was basically a tent.
It was a tent. I mean, it was a storage tent.
There's no windows. Solid walls or
No, we used boxes to segregate our different work areas. So we had a reagent area, we had a PCR area, and we had a post-PCR area that were separated by boxes.
Yeah, I think it's funny because back in 1980, I sequenced the poliovirus genome. It took me 1 year.
It's crazy, isn't it?
1 whole year of just me doing it, running gels. You know, you remember those days? Max M. Gilbert sequencing.
But my only experience of doing sequencing is gel-based sequencing.
Yeah.
Okay, and I spent my entire PhD sequencing 7 kilobases of GC-rich DNA, 200 base pairs at a time, and running them on gels. And now we could do 600 Ebola virus genomes in 5 months.
Technology has just pushed science forward. It's why when I started, you studied 1 virus and 1 isolate, and that was it, because that was all you could do. And now you can study thousands and learn so much more. It's really remarkable. So someone mentioned this morning at the meeting, a lot of the capability that was built in Western Africa just fell apart because there was no more money to keep it going. What's the status of your facility?
Yeah, so that was a concern of ours. The treatment center was closing in September, October 2015. So what we're going to do, you know, we spent $500,000 in bringing all this stuff out.
Yeah.
Now, in reality, the easy thing for me to do would be go home and focus on my norovirus and look after my lab and spend time with my family and do all the. But you know, the right thing to do was to try and figure out how we build some capacity, and we've set up an infectious disease research facility now at the local university. And it's quite a nice. It's quite a nice lab.
We moved the equipment to the university?
We moved all that equipment there. We were able to get more equipment. We were able to find equipment from diagnostic laboratories that were closing down in Sierra Leone, bring it all together.
Nice.
And we've got a nice research facility that is now being used by other researchers. So we heard today about the PREDICT program, which is a USAID funded program to look at reservoirs for Ebola virus. So the laboratory is now one part of that.
Right.
So they're running their
Nice.
Sierra Leone program out of that. Other researchers are coming to use the facility, and we're providing some support to some clinical trials. I'm really interested in the capacity building activity, and that sort of aspect of it. So I'm really keen to try to get people to come and work there and understand that, it is no longer okay just to go in and take samples out of a country and take them home to your own lab.
Right.
You need to come and do something there.
Yeah.
And we have all that equipment. We have ELISA plate machines, sequencers, real-time PCR machines, -80s, everything's there. So you can, we have tissue culture hoods. I have 5 class 2 tissue culture hoods there.
And they have enough funds to maintain all of this and have supplies for it and all of
So
that?
this is the big problem, sustainability. How do you sustain this? So I am only funded until next September. But our hope is if we can engage like-minded individuals who are really interested in doing this sort of research in these environments, we can maintain an income stream. And we want to set the laboratory as a sort of center of excellence for research in that area. And I think I'm hopeful, but then I am quite naive about this stuff. I've never done it before, so I'm learning.
You did quite a bit, it seems. So now, next time, if there's another outbreak in the same area, they will be ready to start looking at samples, right? There won't be this lag when foreign people have to come in and set up shop, and that's really important, right?
It's essential. We've heard a lot today about the issues that are around discovery and trying to identify where these spillover events come from. In my opinion, I think a lot of that money would be much better spent in supporting and developing sustainable programs in these countries. And a lot of money is being poured into this, and it's not straightforward. You can't just put in external funds. The funding has to come from the local governments as well. I think, we can develop all these fancy techniques for identifying pathogens and characterizing them, but at the end of the day, most of the time, all you need is a simple PCR.
Yeah, right.
Nearby the site where the spillover events are occurring.
That's right. So Eva Harris has spent her career doing that in Nicaragua, establishing them to be able to do all of this. And you have to not only get equipment, but you have to have trained people come and train them how to do it. And hopefully it gets sustained. And this is the new face of science, especially in outbreaks and diagnostics. It's not, as you said, taking all the samples back to your country, but doing it right there. And that's how you get the fastest response.
Absolutely. There is a problem with it, though, and it's something I was going to mention tomorrow, and that is that, the academic career structure does not reward the efforts that go into capacity building.
Yes.
If you're a tenure committee and you're assessing two individuals, and you've got somebody who's published a bunch of nice papers and somebody who's invested a lot of time, effort, and money in developing capacity, who's going to get hired?
Yeah.
It's the person who's published all the nice papers.
Of course. Why didn't you bring those samples back to your lab?
Exactly. And then when you think about the individuals who can do capacity building, so people who have tenured posts like me, we can afford to invest that time and effort. If you're a young academic, you just can't do that. You can't spend 3 years setting up a laboratory in a part of Africa or in Brazil, in these low-income countries.
Yeah.
Because it's just not rewarded. So I think we need to change. There's a discussion to be had around how we reward people and for doing this sort of activity and recognize its importance.
Yeah. So when did you end up returning to the UK?
So I was going back and forward. I would generally spend at most 5 weeks in any one time. It was the longest I could stay. I was still having to do my own job back in Cambridge. So
you're head of virology, right?
Well, I'd come back and mock exam scripts, I guess the epidemic was officially over. Well, we thought by the end of 2015. But now we have the research facility. I had been going back and forward now, and actually my last trip was in August, where we've set up an engagement program now where we're teaching high school students essentially about infectious diseases.
Nice.
That's a program we'll be running for the next at
Right.
least 18 months.
There haven't been any cases for many months now, right?
No, there's no more Ebola cases, but of course, you know, in the grand scheme of things, and as somebody quite rightly mentioned yesterday or today, you know, during the Ebola epidemic, more people died of other things than of Ebola.
Right, right.
And, you know, in Sierra Leone at the minute, you know, I think it's 1 in 3 children die before the age of 5. This is huge numbers.
Sure. Yeah, there's a bigger problem that's looming, right? It has to be fixed.
And, you know, I think if we maintain these laboratories and this sort of capacity and local expertise, then we might be able to have an impact.
Yeah. So would you do this again if there were another Ebola outbreak somewhere else in Africa?
Absolutely.
Yeah.
You know, it has been without doubt one of the hardest things, you know, sort of emotionally, physically, and as I've ever done. You know, when you know, we didn't deal with patients, but you would witness, you know, the laboratory basically opened into the red zone. We could see the patients come in, and you could see the children go through the various stages of the disease. So it was a really hard thing to do, but I think it's been one of the most rewarding in terms of, you know, satisfying to feel that, you know, the skills that I've learned over 15, 20 years as a basic biologist. You know, I study proteins and I do biochemistry and, you know, I used to work on poliovirus replication and RNA structures. Some of those skills are actually useful, like really useful for stuff that can, so I would do it again, and I think I would encourage anyone who has the opportunity to do that. I think it really is a very rewarding experience to do.
Well, you're to be commended for doing it and for being persistent at the onset. On TWIV, we had many emails from listeners during the outbreak asking how they could get involved. And, you know, of course, they're not physicians largely, so I'm sure we told them to go to various places, but I'm sure it was hard for them, having less experience than you. But persistence is key, I think, it sounds like.
Absolutely, and I think, you know, I do think there are lessons to be learned about how we tap into the capacity we have in academic laboratories. You know, the people you have in your laboratory, the people, you know, any virologist, anyone who can do PCR could have worked in that epidemic response.
Sure. It's a good thought. I'd never thought of it. I didn't think I would be of any use, but you're right. I can do PCR and set up a lab, right? Basic things, yeah.
You know, and I think some of the skills, you know, just managing, I found that actually just having experience in managing people.
Yes, exactly.
Which is very useful. You know, we're all there. It's a stressed out environment. You know, trying to be able to manage individuals in that environment was quite useful.
Yeah, wonderful story. I've been speaking with Ian Goodfellow of Cambridge University in the UK. Thanks for joining me today.
Thank you.
Great story. I love it. I know we didn't touch on your love, which is noroviruses, but another time we'll talk about noroviruses. I'm Vincent Racaniello from Columbia University, and I'm here for the American Society for Microbiology. Thanks for watching.